nervous system. These are the two major systems for maintaining homeostasis within the body, okay? Is the nervous system, and we'll contrast when we get to the nervous system, contrast hormone systems and the nervous system. They have some things in common, they have some things that are different. So a hormone is a chemical signal, and there's many different types of chemical signals. There's things that are called paracrine and other types of signals. Here, I'm just going to mostly concentrate on hormones, okay? Because there's other types of entities that play chemical roles here. But with a hormone, it's a particular type of chemical signal that's secreted into body fluids. And in vertebrates, of course, that body fluid that it's secreted into is blood in vertebrates. And it really requires just a tiny amount of the hormone in order to bring about a large response. And some of these happen to be neural secretory cells that happen to make and release. I already went over that with ADH. Those are produced by neuron-like cells from the hypothalamus, right? So others happen to be special gland cells that will make and release these. The only cells or tissues in the body that can respond are those that have the appropriate protein receptor. Okay, usually the receptor is always a protein, and depending on the nature of the hormone, whether it's hydrophobic or hydrophilic, that receptor might be, usually, if it's hydrophilic, okay, polar molecule, then that receptor has to be embedded in the plasma membrane with a binding site for the hormone on the outer face of the plasma membrane. If it is lipid soluble, however, the receptor might be in the cytoplasm itself because the hormone can pass through the plasma membrane nonetheless and bind. So there's many types of glands. I'm just going to make two broad categories here. These are exocrine glands. They secrete their products into a duct, a pipeway that will lead to a special place. And some of them we've already come across. For example, exocrine glands would be like the pancreas. The pancreas is both an endocrine and exocrine gland, but for the digestive fluids that happen to be released, the hormone, not necessarily hormones, but digestive material that is secreted into the duodenum, that's still an exocrine gland, and it's there by a pancreatic duct. But as far as the hormones, most of them are endocrine, and so there's no duct. There is a gland, has tissue, and the hormones are secreted out of the cell into interstitial fluids. And of course, what's in the close vicinity of the duct, of the gland, of course, is a capillary, right? And so these endocrine products are crossing the plasma membrane of a endocrine cell into the interstitial fluid, then the endothelium into a capillary, and then they're transported throughout the body by the blood. Okay, now classes of hormones, usually based on their chemical nature. Okay, they classify these based on their chemical nature. So some of these hormones have to be lipid hormones or steroid. Some of them can, not all of them are derived from cholesterol, but a lot of them are. Okay, so aldosterone was a lipid-soluble hormone, and it is derived from cholesterol. Cholesterol is a precursor for it. So you do need cholesterol. Some of these could be fatty acids, okay, in some cases. And some of these hormones are amino acid derivatives, which most of these amino acid derivatives are polar, but not all of them. There's some of them that are non-polar, okay, so that they would operate in a different way than the polar types of hormones. So when I say an amino acid derivative, it is a modified amino acid, or it is an amino acid, okay? Some of these amino acid hormones are amino acids that are not found in the incorporated into proteins, okay? Some of these are peptide or short proteins, okay? We have some of them as well. ADH happens to be a short peptide. When I say peptide, usually talking about a string of amino acids that are somewhere less than 10 amino acid residues or so. Small peptide, or even a glycoprotein. Okay, so belonging to this class, and usually these are polar, and they have to operate through a receptor on the surface of the membrane, also because of their size. Right? So all of these hormones have specific shapes, and therefore they can only be recognized by a receptor on the plasma membrane or inside of the cytoplasm of the target cells that produce that receptor. Usually this receptor is a protein, and due to gene differentiation from one type of tissue to another, some cells don't have those genes turned on, and therefore they don't have the receptors, and therefore they don't respond to the presence of a hormone. Okay, so these hormones, they usually act at very, very low concentrations. Furthermore, some other principles here is that the exact same hormone in one animal may do something very, very different than in another animal. They've evolved different pathways here. An example of this is this is an amino acid derivative, thyroxine. It's produced by the thyroid gland in mammals, and what it does in regulates metabolism, the rate of cellular respiration throughout the body in mammals. But the very same hormone leads to programmed cell death in the tails of tadpoles. So it's involved in the absorption of the tail as the tadpole is metamorphosing into the adult. You can and thyroxine is thyroxine is thyroxine. It doesn't matter whether it comes from a frog or a human, since it's an amino acid derivative. You can take human thyroxine, inject it into a tadpole, and it'll do exactly this. It'll lead to the absorption of the tail. Right, so another feature here of hormones is that even within the same animal, same hormone can have different effects, different responses on different targets. A good example of this is epinephrine. The epinephrine is released from the adrenal gland, and I believe it's a short protein, and it's involved very much in the fight or flight response. Okay, it's very quick, acts real quickly. So if you're under acute stress, okay, someone jumps out at you with a knife or something like that, and you either need to fight or you have to run, one of the way, okay? Nonetheless, your body's physiology has to change very rapidly, okay? So physiological states have to change. And so there's many targets. One of these is liver and skeletal muscle, because these are the places where you store glucose. Most of it in the liver. And so under this stressful condition, you need energy from cellular respiration. You need ATP. And therefore, glucose needs to be mobilized into the bloodstream. And so that's one thing that happens, and it happens real rapidly. Adipose cells, they will also have receptors, and they release fatty acids. Because fatty acids, why? Cellular respiration. Okay, once again, the whole thing here is to increase the supply of ATP for skeletal muscles to respond to this particular emergency. And it can be glucose, it can be fatty acids. Fatty acids will give more ATP. Heart, okay? This glucose and other materials have to bend in oxygen, have to really move quickly to the effectors, the skeletal muscles that are going to respond, and therefore heart rate and volume increase, okay, to move blood quicker throughout the body. Another target, smooth muscles and arterioles. Okay, now this is only in arterioles, not in veins, and especially the arterioles to the skin, not the arterioles to the digestive system or anything else, but arterioles to the skin, they vasoconstrict. Now why do you think that is important? Don't just think of human conditions, but all animals, or even our evolutionary past. This is usually a response to fighting. What happens in a fight? You can get cut. right? You can get cut, start losing blood, all right? So if there's not a lot of blood going to the skin, you're not gonna lose as much. And so this is shunning the blood away from the skin, all right? And another thing here is smooth muscles to arterioles to muscle. They're still smooth muscle. Still smooth muscle, okay, in both cases. They both have receptors to epinephrine, but the response here is vasoconstriction. The response here is vasodilation. Why does this make sense? More blood going, taking oxygen, glucose, other materials necessary to skeletal muscles. Why do you think that, although the receptors are the same, why would be the responses different? Because in that cascade of events within the cell, okay, we have certain kinases in a phosphorylation cascade in that signal transduction pathway. The players are different here than what they are here. Okay, so the internal proteins that are responding to this lead to different outcomes. All right, so very often, okay, so those are, right now I'm just going over some principles, general principles of hormones. And another frequent thing that we'll see with hormones is that they often come in antagonistic pairs or antagonistic systems to maintain homeostasis. And we already saw that with regard to the response to blood pressure in the kidneys. There were two systems there. One, RAS, which responded to low blood pressure, and ANF, which responded to high blood pressure. They're antagonistic. And so this is to maintain a standard homeostatic level of activity. And so something happens here that the activity happens to increase above a certain threshold. Then this stimulates a gland to release a particular hormone, the targets of which respond in such a way to bring the level of physiological level back down into homeostasis level. If, on the other hand, the activity drops below a certain threshold, this triggers usually a totally different organ, a totally different gland to release a totally different hormone that may have similar types of targets or even different targets, but the whole reaction is to bring the biological level back up. And so with the alternating work of these two hormones, it keeps things in homeostatic level. And so this is called antagonistic pairs, and we'll see about that. Now, signal transduction pathways. It's sort of a review, because I did go over signal transduction pathways from Biology 101A. And one of these for non-water is hydrophobic hormones, steroids, sterols, but also the hormone thyroxine. Thyroxine is relatively nonpolar, and therefore these have similar types of effects. Here, their receptor is intracellular. It's inside of the cell, usually in the cytoplasm. And so these hormones can cross. They can cross through the plasma membrane. And usually these are delivered through the blood by escort proteins. And so they're attached to proteins to make them soluble. There's a small amount of the hormone that is soluble itself just in the blood. But as that hormone moves into the cell, then that leads to a change of concentration of the soluble hormone such that more hormones come off of the escort proteins to maintain a constant level. Nonetheless, we get hormones moving in, intracellular, binds to the receptor, and now this here, receptor hormone complex, is a transcription factor. Okay, a special type of transcription factor. And it binds to control regions, not the promoter, but to other control regions. So this is an activator, activating transcription factor. And it'll bind and lead to upgrading of certain genes. So therefore, this will lead to increase in messenger RNA for not just one protein, because a lot of times this activating transcription factor will bind to the enhancers. Okay, remember enhancers for last semester? Enhancers are control regions to which activating transcription factors have to bind. The whole unit has to come around and bind to the promoter before RNA polymerase can bind. Okay, so this here will either turn on a suite of genes that were not active before, or it upregulates the activity of those genes. So we get messenger RNA. That messenger RNA has to move, be brought down to the cytosol, where it links up with the ribosome and translated into a specific protein. And it is the interactions of these proteins that change the behavior of the cell. Okay? Now, one feature about this type of system is that the signal molecules is usually relatively long-lived. In other words, it's not broken down too fast. The cellular response is generally slow, on the order of minutes to hours to days. It really depends on the hormone system, okay? Because it takes time for the activating transcription factor to activate the gene. We have transcription, post-transcriptional modification, then that has to be transported into cytosol, then we get translation, post-translational modification, etc. And then it takes some time. So it's not an immediate thing. It's something that can happen sometimes within minutes, like aldosterone. Probably the activity range there is about 2-3 minutes, okay, but it's not immediate, not like epinephrine. Epinephrine is right away. You can feel it, okay? You can feel yourself with it. So, and generally, so the cellular response is relatively slow, and the duration is somewhat long-term. Okay, here once again, terms talk about minutes to hours to days. Whereas with some of the other hormones I'm going to be talking about, the response is quick, short, and doesn't last long. Okay, but this is with the non-water soluble. And thyroxine, although it is not a steroid, it operates in this fashion too. It changes gene expression. Okay, oh, yes. So aldosterone is one of those. Something that's a little bit longer term are these two hormones in insects. We don't have these hormones, okay? So although I'm mostly going to be concentrating here on mammals, there's still a little bit here that we'll talk about to let you know that other animals also have hormones. So juvenile hormone and ecdysone. The levels of these two hormones, they're both steroid hormones, and they're controlled by certain glands close to the brain. And it's their levels that determine whether a particular molt will lead to another larva, or to a pupa, or to the adult, okay? So as long as juvenile hormone is high, then ecdysone is necessary for the molt, okay? So this becomes high just before they molt, because it's leading, there's a lot of proteins that have to change during molding, okay? That lead to secretion of certain fluids that break down at least part of the exoskeleton, not all of it, and so forth. There's lots of proteins involved here. Ecdysone is involved in that. And this usually molding takes many hours, so it's not right away. Juvenile hormone, its levels are changing. If it stays relatively high, then when this molts, it'll molt into another larva, okay? But if juvenile hormone levels then drop, then the next molt might be into adult, okay? And so as you transform from a larva to an adult, you're talking about a massive number of massive change in gene expression. There's a lot of larval genes that are completely shut off, and a lot of adult genes that have to be activated. And therefore, and this here is long-term, okay? It takes a long time for that to occur. So this is just a figure from your textbook showing cartoon, what's going on here. Hydrophobic hormone, its receptor, this is now, it could be an activator, it could be a repressor. Okay, it really depends, because although I've been emphasizing turning on genes, there's also genes there that you need to turn off. Okay, and so this could be a repressor that binds to the appropriate control region to shut down genes that are no longer needed. Yes.
Do EpiPens work by injecting some kind of hormone?
Say that again.
EpiPens, like when you inject them, do you?
Epinephrine?
Or EpiPens.
EpiPen.
Or like EpiPens, like when you get like the electric.
Oh, yeah, okay, yes. That is decreasing the epinephrine. Yeah, so it's a protagonist of epinephrine. So it's binding the epinephrine and inhibiting it. Okay, so that because that has to be real quick, right? Okay, and then this binds, of course, to the appropriate either enhancer or silencer, but not to the promoter, Okay, now as far as water-soluble hormones, they can't pass through the plasma membrane. Therefore, their receptors have to be transmembrane proteins with binding sites for the hormone on the outer face of the plasma membrane. And so this leads to a signal transduction, so across the membrane of certain players, depending on what the type of system happens to be. And here, the signal is usually very short-lived. Okay, there's things that will break the hormone down real quickly. Okay, so it's short-lived, and furthermore, the cell response is very rapid. So here we're talking about seconds, 2 minutes, instead of minutes to hours. Seconds to minutes can be very, very rapid, and so forth. And usually the response, should have put it up there too, cell response is rapid, but usually short-lived. Usually short-lived, okay? Until the stress, and this is for acute stresses, and then it's over. Okay, so now talking about the types of receptors. Ion channel receptors, ion channel linked receptors. These are like what are in neurons. I'm not going to talk much about them here in this particular lecture. So what these are is that the hormone binds to a receptor that is associated with a ion channel and opens or closes the gate. Okay, and I'm going to reserve that for when we get to neurons, because there's a lot we can say about that there. There's enzyme-linked receptors. Okay, and I probably went over this. It's probably a review also from 101B. Did I cover receptor types in 101 or 101A? You remember that way back, way at the beginning. Sometimes I run out of time and I throw it out, and I don't remember if I threw it out this year or not, or if I went over it. I may have. I may have thrown out some of these and just only went over G-linked receptors, protein linked. But nonetheless, enzyme-linked receptors, the receptor is also the enzyme. And so it functions both as a receptor and an enzyme. And usually it is, not always, but usually the receptor is a kinase that phosphorylates directly targets on the cytosol side. And of course it's leading to the activation of proteins that are already there. Okay, so this is unlike with the steroid hormones where we're changing gene expression, shutting down transcription of certain genes, turning on transcription of other genes. This here, the proteins are already there. And those proteins might be active that we make inactive, or those proteins might be inactive that we make active. But it nonetheless changes the properties of what's happening inside the cell. An example of this, and this is a class. It's not a simple one type of hormone receptor, but there's many different varieties of this. It's called tyrosine kinase receptor. And I'll give an example here. This is from your, notice that this is from chapter 7. Okay, so you'd have to go back to chapter 7 to review this. Tyrosine kinase is a monomer, normally. That means just one unit, transmembrane protein. But if the hormone binds to it, it leads to a conformational change that makes the interior part of the sticky. So that both of these, we have two of them, they're floating around. Normally, they don't stick together, but if they bind a hormone, the appropriate hormone, they dimerize. Okay, dimerize, they become two of them bound together. That leads to yet further conformational changes, which activates a kinase, ATP kinase, that will phosphorylate the tyrosines. There's 6 different tyrosines on the catalytic region of this protein that will become bound to phosphate, which further leads to its activation. Now it will then phosphorylate other signaling proteins. And usually it's branched. These phosphorylation cascade will, have 2 different branches to it, and it gets to be real complicated, all right? So this is how it's working. It's leading to 2 different cellular responses. And usually the cascade is in this from, there's an eraser. First of all, there's something called mitogen activated protein kinase 1, which will phosphorylate an inactive mitogen activated protein kinase 2, which then will phosphorylate mitogen activated protein kinase 3, which then phosphorylates something else. This type of thing is very common with a cascade in tyrosine kinase types of receptors. What is more common than tyrosine kinase types of receptors is G protein linked receptors. And I just went over one of those not too long ago. So here, the G protein, the receptor and the effector proteins are different. In other words, the catalytic enzyme is separate from the receptor. That's not the style of tyrosine kinase receptors. It is both the receptor and the effector. So here we have a receptor, binds, and associated with it is a G protein. G protein family is a huge family, okay, huge family. And in fact, some of that, a certain type of G protein is involved upstream of this cascade system here, tyrosine kinase. It's called RAS. That's just a monomer that's involved there. You don't need to worry too much about that. But nonetheless, this particular G protein is a trimer, 3 subunits, and what activates the protein is not phosphorylation, but whether or not it's bound to a particular nucleotide derivative, guanosine triphosphate or guanosine diphosphate. If it is bound to binding site has guanosine diphosphate, then the relay protein, the G protein, is inactive. And so what happens is that it will exchange. This is not phosphorylation, but when it is stimulated by the receptor, then it changes conformation such that the binding site no longer can bind GDP. That leaves, and what can enter there now is GTP. This is an exchange. No phosphorylations happen. It's just an exchange of one for the other. And this is what activates the G protein. And one of the subunits of the G protein then will dissociate and translocate along the inner face of the plasma membrane until it runs into a binding site on an effector protein. This is the enzyme. And there's different effector proteins. I'm only going to talk about one, but this will lead to the production of a secondary messenger that's on the cytoplasmic side. And the secondary messengers can be different, okay, from one system to another. And what the secondary messenger does is it binds to an inactive protein and activates it and starts a phosphorylation cascade. And I'll illustrate this with a figure on the next slide. first of all, wait for everyone to get this. So all these are activated by, so here we have a receptor, we have a stimulus, and I probably showed this in 101A. Binds to the receptor, leads to conformational change in the receptor, which leads to a conformational change in G protein. And what I'm not showing here is the exchange of GDP for GTP that happens here. And that leads to, and this is the one that has the binding site. So the subunit that has the binding site for the GTP dissociates, binds to an effector. In this case, it's adenylate cyclase. Okay, there's others. Okay, depends. They do something else. What adenylate cyclase does, it is an enzyme that converts ATP into cyclic AMP. Okay, so it converts cyclic AMP into, or ATP and cyclic AMP, and this is the second messenger. So the hormone did not cross through the membrane, but now we have an increase of cyclic AMP on the cytoplasm side, cytosol side. And what this binds to, there are inactive proteins called cyclic AMP dependent protein kinase. It has to bind to the binding site. Only those proteins that have the binding site for cyclic AMP will respond. This binds, leads to a conformational change, which then, let's leave that out for now, which then leads to the phosphorylation cascade. Because this is a kinase, its target is other proteins that are phosphorylated to activate it. And we went over those phosphorylation cascades last semester. Now to shut this down, if in some cases it requires an antagonistic hormone, one that's antagonistic to the stimulatory hormone. And it's a different, totally different receptor. It binds, and it's a different type of G protein, because there's several different forms of G protein. But then this leads to that binding to adenylate cyclase, which will break it down. Okay? After that, as long as this is still there, then this continues to be stimulated. So we got to get rid of that. And so something that is constitutive, always there, is cyclic AMP phosphodiesterase. It is always there in the cell, always active. So it is constantly breaking cyclic AMP down into simply AMP. It's a little difference, but it's a big difference as far as these proteins are concerned, because simple AMP cannot bind to the cyclic AMP dependent protein kinase. And so as this cyclic AMP is broken down, cyclic AMP is removed from these, and the system is shut down. Now, why do you think, and I even asked this question in 101A, if this cyclic AMP phosphodiesterase is always there, always working, why would producing cyclic AMP lead to activation? It's always there breaking this down.
For cyclic AMP, it can.
Well, this here is producing high amounts of cyclic AMP, higher than what the cyclic AMP phosphodiesterase can handle. And therefore, it can't break it down fast enough. And that leads to the activation of this. Now, once we shut this down, once it is shut down, then this here can recover, okay? But it's always there, always working. So this system happens real fast, and it can be shut down real fast. Now, the whole point of this, okay, this is just the figure, once again, of ATP, adenylate cyclase will convert it into cyclic AMP. Phosphate has a double, that's why it's called cyclic, between carbon number 5, carbon number 3, and phosphate. And what the cyclic AMP phosphodiesterase does, it hydrolyzes this link right over here, so that it's now just AMP. Although these look similar, a little difference makes a big difference. They don't stimulate the same. Okay, this is where I'm going to stop, about that time. So I will continue with this on Thursday from slide 15. Then we'll get into some of the specific hormones, once I've covered the major ways in which hormones in general act. Okay, that's it. Yes,
Will Monday's quiz start from today's lecture?
from today's lecture.
Oh.
Yeah, so it would be from the kidneys, from mammalian kidneys. Since I reviewed all of the pathways of anatomy, everything, then it's going to, the mammalian parts that cover the mammalian kidney will be starting next year.
Okay, so like from